This video abstract is AI generated. What makes one part of a protein surface easy to reach, while another region remains protected? Protein recognition is often described in terms of shape. If a ligand fits into a pocket, we call that region accessible. But this manuscript asks a deeper question: is surface accessibility controlled only by geometry, or also by the water molecules that surround the protein? To test this idea, the study used α-bungarotoxin as a model system. Alpha-bungarotoxin is a small, well-characterized neurotoxin with a 3-finger fold and a known receptor binding site. Because its structure and binding surface were already known, it was an ideal system to ask how hydration controls molecular access. The first step was molecular dynamics. Simulations mapped where water molecules remain close to the toxin surface. Many stable hydration sites were found across the globular core and on fingers 1 and 3. But the receptor binding site was different. This functional region had few stable waters, suggesting that it is not protected by a rigid hydration shell. The next step was NMR epoxy experiments detected protein protons that are close to long residence water molecules. These measurements supported the hydration pattern predicted by molecular dynamics. Together, MD and NMR produced a validated map of the toxin hydration shell. The authors then asked whether this hydration map predicts surface accessibility. They added the soluble paramagnetic probe gadolinium DTP-ABM and measured attenuation in proton-carbon HSQC spectra. Where the probe approached the protein frequently, NMR signals were strongly attenuated. Where the surface was shielded, attenuation was weaker. The key result was that accessibility did not simply follow geometric exposure. Some surface-exposed regions were poorly accessible because they were protected by stable waters. In contrast, glycine 37, located in the poorly hydrated binding site region, showed very strong paramagnetic attenuation, even though simple exposure alone would not predict it. This leads to the central model. The solvation shell acts as a gatekeeper. Stable, long-residence water molecules can shield a protein surface. Poorly hydrated, dynamic regions are easier for outside molecules to approach and become surface hot spots for recognition. In this view, protein surface accessibility emerges from structure, dynamics, and hydration together. The combination of MD, hydration-sensitive NMR, and paramagnetic probes provides a general strategy to identify functional surface regions. To know more, read our work online.